My name is Mary Ford, and I'm the Director of Engagement and External Relations for Maricus, and also the co-coordinator for MACAN. This is the last scheduled webinar in our fourth MACAN webinar series. And today we're excited to host our two co-presenters, Drs. Emily Osborne and Shannon Misek, on the NOAA Ocean and Great Lakes Acidification Research Plan. Dr. Emily Osborne is a scientist at NOAA and currently works within the research branch for the Ocean Acidification Program. Since joining NOAA in 2017, she's served as an editor and author for several international and national climate assessments, worked to develop an agency-wide and interagency research strategies, and published peer-reviewed original research on ocean and climate science. Emily earned her PhD in marine science from the University of South Carolina in 2016 and a BS in geology from the College of Charleston in 2012. Her research expertise is in developing paleoproxies using the fossil record on the seafloor to understand ocean conditions of the past, but her scientific interests span a broad range of global change issues. Dr. Shannon Misek is a research scientist at the NOAA National Marine Fisheries Laboratory in Milford, Connecticut. Since joining NOAA in 2002, she served as a research scientist examining environmental effects on marine bivalves. She started working on how ocean acidification affects a variety of marine organisms, including phytoplankton, bivalves, and fish. She's worked to develop agency-wide and interagency research strategies and published peer-reviewed original research on ocean and climate science. Shannon earned her PhD in oceanography from Old Dominion University in 2002 and a BS in environmental science chemistry from SUNY Plattsburgh in 1997. We have about 20 minutes at the end of the presentation for any questions and comments. Just a reminder, I know everybody's been on a million of these, but all participants will be muted. So ask your questions via the question box. Anthony Himes, a PhD student at the Virginia Institute of Marine Science and the Virginia Sea Grant Fellow assisting with MACAN efforts, will be collating the questions to ask at the end of the presentation. So feel free to submit your questions at any time you think of them during the presentation. Just a quick reminder, the webinar is being recorded and will be available on the MACAN website section with our previous webinars in the series. So with that, I'll pass it off to Emily and Shannon.
Thanks, Mary. This is Emily. I'm starting to show my screen. Can you guys see my slides okay? Anybody?
Yes, we can.
Okay, awesome. Thanks, Anthony. So thanks so much for having us here. Good morning, everyone. We're looking forward to talking with you about the NOAA Ocean and Great Lakes Acidification Research Plan. The plan we'll be talking about today will span the years 2020 to 2029. You guys are getting an early look into the plan as it has not. We're in the final stages of copy editing and typesetting the plan and anticipate it coming out at some point this summer, so in the next month or two. So I served as a coordinating editor for the research plan, and Shannon served as a lead author and focused on developing a regional plan, which she'll be talking about in the second half of our presentation today. So to provide a little bit of context, this is NOAA's second Ocean and Great Lakes Acidification Research Plan. The first plan was released in 2010 and has been guiding the research that the agency has been conducting over the last decade. So much of the work that has gone into the 2020 to 29 plan is reflective of some of the foundational science that the 2010 plan put forward. But again, we're really focusing on building upon the knowledge that's been developed over the last decade and turning our attention to some new issues that the agency hopes to tackle in the coming decade. So to give you a little bit of background on the way that the plan is organized, I have a short table of contents here. The plan opens with an introduction, which provides a little bit of context for why NOAA conducts ocean and Great Lakes acidification research, what some of our legislative mandates have been, how the agency is structured, and how we tackle different types of ocean acidification science. I do want to emphasize that this is an agency-wide plan, so it crosses all of NOAA's line offices. We have many authors and coordinating editors from each of the NOAA line offices. So it kind of breaks apart which line office is responsible for which aspect of NOAA's OA work. And then from there, the plan opens with a national research plan, which I will be talking about with you today and in the coming slides, which really focuses on our high-level research objectives with respect to ocean and Great Lakes acidification science. So it pulls upon all of the regional needs and kind of provides just a high-level view of what the agency hopes to focus on in the coming decade. From there, there's a series of regional research plans. So we have standalone regional chapters for the open ocean. So any space beyond the continental shelf, we define as open ocean, as well as an Alaska and Arctic chapter, which previously in the 2010 plan had been lumped into a single chapter. This time around, we decided to break them apart. So we have standalone chapters for each of those regions, as well as the West Coast, the US Pacific Islands, Southeast and Gulf of Mexico, Caribbean and the Florida Keys, the Mid-Atlantic Bight and New England chapters, which again had previously been lumped together as one region. Shannon was a lead on both of those. We'll be talking in greater detail what both of those regional plans contain in the coming slides as well. And last but not least, we also have a Great Lakes acidification research plan as well. So the way that we decided to approach each of the research plans, so all of the regional plans, the open ocean plan, as well as the national plan, all take a similar approach in how they're structured. So we have 3 different themes that each of those plans focus on that you can see here on the screen. We fondly refer to it as our trifecta. So the trifecta focuses on observing what's going on in the environment through OA monitoring and modeling research. We also aim to look at the biological sensitivity, or looking at the response of organisms and the development of ecosystem level models, as well as human dimensions, which focuses in on socio-economic impacts, education and outreach, as well as adaptation strategies and management support. So just to kind of drive this point home, because it's a really important kind of theme that runs throughout the entire plan that we're releasing, is that we're really focusing on these 3 different activity areas. So documenting and predicting environmental change via monitoring, analysis, and modeling, characterizing and predicting biological sensitivity of both species as well as ecosystems as a whole, and understanding human dimensions and socioeconomic impacts of this environmental change and biological sensitivity. So collectively, the OA trifecta, which we hope to get at a better understanding of the vulnerability of US communities and economies to ocean acidification. So now diving into the research objectives that are featured in the national chapter, we again have 3 research objectives that kind of neatly fit within the trifecta that we were just talking about. The first one is to expand and advance OA observing systems and technologies in order to improve our understanding as well as our predictive capability to model OA trends as well as processes. This is both on a global as well as a regional basis. The second objective focuses on the biological sensitivity focal area, which is to understand and predict ecosystem response, as well as the adaptive capacity of ecologically and economically important species to OA and co-stressors. There's a strong focus here in this research objective to really hone in on the way that OA is progressing in tandem with other stressors. So understanding biological response in the framework of a multi-stressor environmental system. So understanding how temperature, oxygenation, eutrophication, a number of processes are acting together with OA to stress ecosystems. And last is our human dimensions focal area research objective, which is to both identify and engage OA stakeholders and partners to assess their needs and generate products that are responsive to those needs, as well as tools that can support management, adaptation, and resilience to ocean acidification. So I want to take the next few minutes to just dive a little bit deeper into each one of these objectives, because each of these objectives has a series of research actions that are associated with them. So I'd just like to provide you a little bit of detail on kind of more of the finer details within each of these research objectives. So each research objective within the plan has a rationale that's laid out. We've kind of gone over some of the details as to why we hope to focus on this objective, so I'm not going to spend much time on this slide. Instead, I want to go through the actions that are nested underneath research objective 1, which is to develop cost-effective and robust systems, sensors, and technology, and transition these new tools as operational elements of the OA observing system. So just a really foundational improvement of our OA observing technologies. Action 2 is to collect carbonate chemistry parameters with the purpose of constraining specifically the anthropogenic carbon content within the open ocean as well as coastal environments to really understand the human fingerprint within the regions. Action 3 is to improve the leveraging of remote sensing, specifically satellite products, to upscale our in-situ observations in an effort to produce estimates of global surface ocean carbonate chemistry, getting a better synoptic view of what's going on by using our satellite tools. Action 4 is to strategically deploy observing system assets for optimal spatial and temporal coverage based on regional and global modeling needs. So this just means laying out our research questions, for example, on a regional basis, and really understanding what exact observations we need in order to answer those questions and strategically deploying them as we are limited in how much observing we're able to do. Action 5 is to develop and expand coverage of biogeochemical and ecosystem linked models, especially where we have relevant marine resources and communities that are dependent on those marine resources. Action 6 is to continue leadership and support of the enhancement of the Global OA Observing Network, or GOA-OM, within which the NOAA OA Observing System is nested. If you're not familiar with what the GOA-OM is, it's essentially an international organization of countries that come together to work to develop a global OA observing system that NOAA has been one of the leaders of. Action 7 is to ensure that data management plans are based on standard formats and also comply with FAIR data principles, so they're easily accessible. And Action 8 is to support data synthesis to ensure that those data and observations that are collected are then transitioned into useful products that can be ingested by modelers and other audiences for further research and analysis. Okay. So moving into our second research objective, again, not going to spend too much time on this slide, but this objective, as a reminder, was to understand and predict ecosystem response and adaptive capacity of ecologically and economically important species to OA as well as co-stressors, so really focusing on that multi-stressor framework. Action 1 is to assess OA and a multi-stressor sensitivity among species, particularly ecologically and economically important species, as that is a mandate of NOAA as an agency to focus on those species. Action 2 is to co-locate physical, chemical, and biological observing systems in order to improve our understanding of multi-stressor interactions. So in some cases, this might be moving observing assets that might currently exist in order to better get a suite of observations of physical, chemical, and biological data to get at this question. Action 3 is characterize the full marine carbonate system rather than, for example, just measuring a single parameter. in order to support attribution science of species physiological response. So this is to say that really understanding how the variables of the marine carbonate system kind of independently are changing, we can get a better understanding of what specific variables might be influencing an organism. Action 4 is foster new OA research to study emerging and unanticipated ecosystem changes. We put this in there because we've seen in the last couple of years and have been turning our attention to try to understand the relation between ocean acidification and harmful algal blooms. So remaining prepared to kind of turn our attention and research those emerging and unanticipated environmental changes was important. Action 5, use new biological sensitivity knowledge that we've generated as a part of actions 1 through 3 to improve our ecosystem and biogeochemical models that we can then use to project future change within these ecosystems. Action 6, assess sensitivity and evaluate the potential for biological capacity to adapt and acclimate to OA and multi-stressor environments. So this is looking at the genetic signature and individual responses of some organisms within populations, et cetera. So kind of an exciting new field of research. In Action 7, identifying species resistance and resilience to support mitigation strategies, foster resilient communities, and enhance habitat restoration. So this is really building upon Action 6, taking that knowledge and applying it in order to kind of respond to the change that these organisms and ecosystems are experiencing. And the last research objective, as a reminder, was focused on our human dimensions research area, which was to identify and engage stakeholders and partners to assess their needs and generate products and tools that support management, adaptation, and resilience to OA. And those research actions are 1, to identify and build relationships with those vulnerable communities, stakeholders, and partners, to identify their needs and concerns and exchange knowledge with them. Action 2, develop a strategy for engaging Indigenous governments and communities in order to better exchange knowledge and integrate Indigenous and scientific knowledge together to ultimately produce a better understanding and product. Action 3, model economic, cultural, and social impacts to evaluate intervention actions and also explore adaptive strategies that can build resilience in these communities. Action 4 is to encourage research partners and two-way dialogues with stakeholders to ensure that science is aligned with the local and regional level priorities. So really gaining a better understanding of where OA kind of nests within their environmental concerns that their communities are facing. Action 5, develop and operationalize data synthesis and visualization tools, as well as communication products with robust input from our stakeholders and partners to ensure that those products are responsive to their needs. Action 6, create education and outreach resources based on the regional needs in order to promote an understanding and awareness of OA and those resilience strategies within those regions. And also Action 7, monitor the trends and community awareness and perceptions of OA and impacts to participation and stewardship activities across our diverse stakeholder groups. And with that, that wraps up our summary for the national chapter, and we're going to shift our attention to our regional chapters, which Shannon led on the Mid-Atlantic region. So I'll hand it over to you, Shannon.
Okay, I assume everyone can see my screen now. Yes. With the 2010 planning document, as Emily mentioned, the Mid-Atlantic and the Gulf of Maine were considered one region, and it was the Northeast region. In the new planning document, it has been split into 2 regions. So we have the Gulf of Maine as one chapter, and we have the Mid-Atlantic region as a separate chapter. And this was because of the different dynamics between the 2 regions, and we felt it was better to separate the 2. I'd like to, before I begin, I'd like to thank all the authors that ended up working on this chapter, which was Chris Kincaid, Chris Chambers, Chris Melrose, Kim Hyde, Beth Phelan, Beth Turner, and Matt Poach. All of them played a critical role in writing this chapter and bringing it together. So, why can I not? There we go. Okay, so the Mid-Atlantic region in this new document is defined as the area south of Cape Cod, Massachusetts, to Cape Hatteras. It includes the large continental shelf. It includes the multiple shelf breaks that are located there, 5 estuarine ecosystems that play a critical role in the dynamics at the in the coastal zones, and it has lots of barrier islands within the area. We have cold polar water coming from the Labrador Current that comes from the north down, and we have the warm Gulf Stream water that comes from the south up. And these areas tend to have different temperatures, and these water masses have different temperatures and different carbonate chemistry. From in the region, as you know, we also have the fall-winter mixing events that get rid of the thermocline that usually develops in the late spring, early summer, and that lasts until we get the mixing events. There's a really large cold pool located off the coastal zone where there's a lot of economic and recreational fishing that occurs, and the water temperature, as you can see, tends to be much lower in the cold pool relative to the coastal zones. And there can be upwelling throughout the coastal zones here. The region has been defined as being highly susceptible to ocean acidification, especially areas within the Chesapeake Bay and off the coast of New Jersey and Delaware. It also has been identified as a region where eutrophication may contribute to the OA signal. So between the two, high social vulnerability and eutrophication, and the economic value in the region, understanding the drivers and the what's going on is important. And so we try to highlight that in this plan. So as Emily said, we have the trifecta even in the regional chapters, looking at the environmental, looking at the monitoring and modeling, looking at the biological response, and then looking at the human dimension, and trying to combine them and come up with a cohesive plan. So for the Mid-Atlantic regional chapter, what we know so far from the monitoring that's been going on is that we have very strong seasonal variability. As you move from the north down to the south towards Cape Hatteras, there's a gradient with more acidic water in the northern reach and less acidic water in the southern reach. You have the cold Labrador Current coming down, and you have the Gulf Stream going up. So we get this north-south gradient. We also get a gradient from the coastal zone to the open ocean. And we do have some bottom water surveys that suggest that with the stratification, you do get a different signal in the carbonate chemistry, and you can pick up some of the primary productivity that goes on in that area. So with what we already know, we developed what the next 10 years from 2020 to 2029 would look like and what to focus on. So one of the first objectives was to improve OA forecasting across daily to decadal timescales by modifying regional observing systems that better quantify the drivers and looking at the vertical resolution of the carbonate chemistry dynamics and looking at the interfaces between them. And so the first action item is doing carbonate chemistry measurements coupled with other environmental parameters, and to really examine what is going on from the surface to the benthos and from the estuaries to the open ocean. The second action item is to synthesize the data to understand the carbonate chemistry dynamics of the different water masses and different sources and the temporal changes that occur in the mid-Atlantic bight. The third action item was to promote, synthesize, and coordinate the riverine input to the estuaries to determine how river discharge affects alkalinity and ocean acidification within the mid-Atlantic bight region and into the coastal zones. The mid-Atlantic region is experiencing increased precipitation with climate change. Last year in the Chesapeake Bay and other estuaries, due to the high river discharge from rain events, alkalinity was driven down, and so was salinity in a lot of areas. And so to better assess how those drivers are going to affect carbonate chemistry was really the goal of this action item. The fourth action item was to use new technologies to better assess the relative contribution of upwelling, hypoxia, nutrients, and sediment load on OA in the region. And all of this was to improve the OA forecasting on daily to decadal time scales. The second action item was to simulate the full water column carbonate chemistry dynamics of shelf and primary estuarine systems. And this would be accomplished by collating and synthesizing existing carbonate chemistry data in the region that can be used for model validation. The second was to actually continue the development of biogeochemical models to characterize ocean acidification and understand what mechanisms are driving the conditions that we're observing. The continued development of biogeochemical regional models like ROMs to include other variables like temperature, oxygen levels, and eutrophication was the third action item. And the last one was to conduct studies to inform biogeochemical models to evaluate the dynamics at the sediment water interface with increased OA, eutrophication, and hypoxia. And this last action item had a lot to do with marine organisms living at the sediment water interface and trying to understand what is going on in the environment that they're living in, especially bivalves and crabs that are at the sediment. So then we moved into the biological sensitivity, and what we know about the region is that there's an increase in water temperature at a rate greater than anywhere else in the globe. We also know that it is a region that is faced with high eutrophication. The commercial species of oyster and surf clams and sea scallops is really important for the economy in the mid-Atlantic bight, and you also have blue crabs and sturgeon and flounder. Of the, there was an assessment on the Northeast continental shelf by Hare et al., and of the species that are within the mid-Atlantic bight, 27% of the taxa were considered highly vulnerable to climate change. And of those species, there are still 69% of them that have not had any research on ocean acidification done. So this section was trying to look at those questions. So the first research objective was to determine how OA and other multi-stressors impact ecological or economically important marine species. So the first action item to do this was to develop experiments to address population and life stage response with respect to OA environmental stressors for important shellfish, crustacean, and finfish in this area. So this would include looking at what goes on with eutrophication, hypoxia, temperature increases, and looking at the population and life stage response. Then second action was to characterize phenotype plasticity and genetic potential to adapt to OA and other environmental stresses that may be occurring. The third action item was to use experimental physiological and mechanistic measures to determine the energetic cost of acclimating to ocean acidification. And the last action item was to encourage field experiments that use existing platforms like hatcheries and restored oyster reefs to monitor the physiological and life stage response. This would include direct and indirect observations as needed. The second biological action was to use experimental results to parameterize dynamic process models that allow the evaluation of within and among generational consequences of ocean acidification. So there is a strong link between experimental efforts with population ecosystem models to identify and rank the highest value information at appropriate scales. The second action was to ground truth models with experimental testing of predictions within and beyond the parameter framework of the model. And the last action was to compare and contrast models for sensitivity and robustness within the Mid-Atlantic Bight region. So then we look at the human dimension. In the Mid-Atlantic region, commercial fishing was valued over $8 million, and recreational tourism fishing was at $3.5 billion. The area is highly dependent on shellfish species. In Virginia, it is the largest exporter of the eastern oyster and harvests a lot of clams in the United States. Aquaculture is expanding in the region from North Carolina on up to Massachusetts. And as we promote the blue economy, there's been an increase in aquaculture. So understanding the human dimension and assessing the risk and vulnerabilities within the human dimension is an important goal in this chapter. So the first objective to do this was understand how OA will impact fish harvest, aquaculture, and communities. So to achieve this, we have the first action item as expanding the observation capacities at aquaculture sites by including hatcheries and shellfish farms as OA monitoring sites to better understand the OA drivers on the local scale. The second was to expand model capacities to use species-specific data to predict economic impacts. The third action item was models to include OA conditions combined with eutrophication and hypoxia. And the last action was to create habitat suitability maps that will give us an estimate when time thresholds are made with the carbonate chemistry, when it will potentially be no longer profitable to continue growing or harvesting shellfish. The second research objective under human dimension was to evaluate the benefits and cost of mitigation and adaptation strategies. So the first action was to determine the cost of mitigation strategies in fisheries relocating due to displacement by ocean acidification. The second action was to determine, are there specific strains or breeds of species that are better able to respond to OA condition? Is there some type of genetic hardening going on between species that could be identified? And the third action was alternative management options to ensure maximum sustainable fisheries yield and aquaculture production under future conditions. And the last objective under human dimension was to integrate OA understanding into regional planning and management. And this action involved conducting comprehensive management strategy evaluation and scenario development to assess the ability of fisheries management to react to changes in harvest populations. To support economic modeling in sociological studies to determine the ability of fishery members in aquacultures to alter practices due to changes. To develop climate-induced social vulnerability indices with respect to OA to improve the understanding of how communities might respond to OA in a resilient way. And the last action item was to incorporate OA research findings into existing NOAA products and support management, such as the National Marine Fisheries Ecosystem Status Reports. And this was the highlight of the entire Mid-Atlantic Regional Chapter, trying to bring together the three components of monitoring, biological response, and human dimension. And that is it. Thank you.
Thank you, Emily, and thank you, Shannon. That was a lot of really great information. With that, I'll just remind everybody, if you have any questions, please type them into the questions box. I think we have a couple so far, so I'll pass it over to Anthony.
Yeah, thanks, Mary. We do have a few that have come in so far, but feel free to keep sending them in. Our first question, I think both Emily and Shannon, you can both speak to this, but the actions that are under each research objective, have they been prioritized in any way? And if so, how is that determined?
So this is Emily. Yes and no. So recently the Ocean Acidification Program had a meeting in January where we got together all of our principal and guest investigator researchers, and we took a hard look at the research plan and took an opportunity to revise some of at least the wording of the research objectives, as well as some of the actions. During that meeting, we talked about what are the kind of near-term actions that we need to focus our attention on, kind of trying to avoid using the word priority, because it's hard to say what is more important. When you look across the actions, they're all things that we really hope to achieve. But in terms of what things need to happen first, we have talked about that. It's not reflected in the research plan. It's reflected in some of the internal program plans that we have. But the numbering of the actions under the research objectives are somewhat arbitrary, at least for the national chapter. And I can turn over to Shannon to see kind of her thoughts on the chapters that she led.
Yes, so I led both I led the New England chapter, and I was a co-author on the Mid-Atlantic chapter. And it wasn't by priority. We definitely did not number them by priority. So I'm just restating what Emily's saying. So there's no real priority in the way that they are numbered.
Okay, our next question, first is a comment regarding that there's conflict in the literature regarding how ocean acidification impacts animal behavior. And the question is NOAA going to pursue research on animal behavior to investigate and try and resolve this controversy?
So there is focus in this research plan to not only look at, I guess, historically what has been most heavily studied in OA science, which are our marine calcifiers, but also those higher trophic organisms. We've even talked at the January meeting that I was just mentioning about understanding the impact that OA could have on whales. So yes, that is a priority to conduct some of that foundational research to understand, and that is happening right now at NOAA as well. Some of those behavioral impacts as well.
And I'd just like to follow up that the document, originally when the document, certain life stages were studied, and the document really calls for all life stages to be studied, because a lot of the research out there has shown that at different life stages, you do get different responses. And that's why, if you notice in the Mid-Atlantic region, it says population and genetics and plasticity, to try to highlight some of those research results that are showing that different life stages do result in slightly different behaviors.
Okay, our next question is, when will the report be available?
Great question. So we had hoped to release it prior to today. There have been some delays. It's a really big author team. There are more than 80 authors that are contributing to this report, and we've taken a lot of stops along the way to make sure that we're getting input, which has been really valuable. And so we're not upset that our timeline has been extended a bit. But right now we are shooting for the middle of this summer, so sometime within the time frame, hopefully of July, and really August at the latest. But we are pushing as hard as we can to get it out as quickly as possible.
We have another question here. One of the actions mentioned developing new technologies for water quality monitoring. How is there any sort of timetable or effort currently being conducted to advance new technologies, as they would greatly impact research on other action items?
I'm not sure I fully understand that question, but the technology piece is something that's been underway at NOAA for a while now. It wasn't, I think, super highlighted in the 2010 research plan, but has certainly been an effort that we've been working on over the last several years, and really wanted to bring it to the top, since observing by ships exclusively isn't really helping us to get at the best kind of sense seasonally or spatially what's going on. So there are a number of independent, groups and researchers that are developing these technologies. And I think, Anthony, I might not be answering the second half of the question about how it's impacting. What was the?
I think the main drive of the question is, with one of the action items being developing new technologies, a lot of, say, new technologies in water quality monitoring would advance kind of the action items under the environmental portion of your trifecta. So is there any kind of initiative really pushing for those developments, as it will aid other components of the plan, if you're aware of any like that?
I don't know if there maybe is like a formal group that's thinking about like what's our timeline and what kind of questions are we trying to tackle. But I know that like, for example, at PMEL, there's an engineering group that is really linked in with the researchers, the carbonate chemistry scientists, and trying to respond to their questions and needs. But that's maybe something for NOAA to consider, to pull together kind of the engineering side of the house and the researchers more formally to get at those types of questions.
Okay, I have one more question here, but if anybody else wants to send one in, feel free to do that now. So this plan has been developed 10 years after the previous plan. Is that kind of the plan going forward, every 10 years that these will be updated or revised, or has that been thought about at all?
Yeah, so the goal moving forward would be, again, to revise this massive research plan in another 10 years. We wrote it with the intention of trying to keep it open-ended and applicable once we get to the year 2029, by including some of those actions that are like anticipate emerging or unanticipated events, researching those types of things. But with that said, yes, this is a every 10-year type effort. This was a major undertaking. It took almost, actually more than a year to pull together this research plan, just because the nature of it pulling together all of the different components across the agency and authors and experts and getting the proper input is quite a bit of work. So not planning on doing it again until our next decade is upon us.
Okay, I don't see any other last minute questions. So thank you very much for your answers to those. And with that, I'll turn it back over to you, Mary.
Thanks, Anthony. And thanks again to Emily and Shannon. It was a really great presentation and great answers to the questions. Thanks for everyone for joining us on today's webinar and through the whole webinar series. Just another reminder that a recording will be posted on midacan.org, and we'll be in touch when the next webinar series is scheduled. I hope everyone has a great day, and we look forward to talking with you all soon.
Thanks for having us, Mary.
Bye.